Hi, I'm Oviya Kalaivanan,
And I'm Aiden Kurtana.
and this is the Think Thesis podcast. Here we have our guest today, Oviya Kalaivanan. Miss Kalaivanan, do you want to introduce yourself?
Sure. Thank you guys for having me today. My name is Oviya. I'm a rising senior at Washington University in St. Louis, studying cognitive neuroscience and minoring in anthropology, global health, and environment. I'm super excited to be here. I've been involved in research for about 3 years now. I started my freshman year of college. It's about to come to an end, which I'm really sad about, but I'm really happy to be here and share my experience getting into research.
Well, thank you so much for being here.
Yeah,
Yeah, you're our first guest.
that's exciting.
Hooray!
All right, I'm gonna go ahead and start. What initially sparked your interest in this research topic?
Sure. So I study Huntington's disease. So I look at the molecular biology behind that disease. And growing up, my grandmother actually had Huntington's disease. So kind of seeing that in the family sparked that interest in me. Like seeing her decline, I felt really helpless and I really wanted to step in and do something. So going into college, I'm like, how do I make a difference in this field? And I realized maybe research could be an avenue for me to contribute to Huntington's disease research. So my freshman year, I kind of went on Google, did like a Google search, and kind of saw what labs in my vicinity, probably in my university, and there's a couple down the road, to see what universities offered labs related to Huntington's disease or like in general motor diseases. And I found out one of the labs was pretty close by, called the Yano lab, and I sent a bunch of cold emails. So that wasn't the only lab I contacted, so it took a lot of time. So about 50 emails to like labs from, I don't know, nutrition to labs ranging to cancer. And I feel like through the interview, so I had an interview with my lab, like her mission really stuck out to me. I really resonated with it. And I really wanted to give back to the HD community. So I decided to go with that and explore Huntington's disease and specifically the protein interactions that cause neurodegeneration in HD.
Mhm. So, would you mind
So,
walking us through the problems that you're trying to address with your research?
Sure. So my project basically focuses on understanding the interaction between a DNA methyltransferase, which is basically a protein, and I wanted to see what other proteins bind to it to kind of cause the neurodegeneration in neurons. So just for like context for our viewers, so what is
Mm.
Huntington's disease? So it's kind of this devastating neurodegenerative disorder, and it leads to progressive neuron dysfunction. So we don't know what exactly causes this disease. So we just know that this dysfunction basically leads to abnormal polyglutamine repeats. So a person with HD has about 45 or more repeats, which kind of causes them to get HD. So our lab is trying to find out what protein interaction kind of causes these abnormal amount of polyglutamine repeats. So that's what we kind of work on. And I think our lab had already identified all the different proteins that interact with this DNM3TA, which is the DNA methyltransferase. And when I came in, they wanted me to work on one specific protein to see if the interaction was there and if that interaction caused the neurodegeneration in neurons. So that's kind of like the big problem that I've been working on for 3 years. Obviously, since there's no cure and since there's no real reason as to why this occurs, this research has been really time consuming.
Mhm.
It requires a lot of patience. So I've been trying to, try different experiments because this hasn't been done before to see why this interaction is important or if this interaction is important. We still don't know. But yeah, that's kind of like the high level view of my work.
Mhm.
yeah, and you talked about Huntington's disease and how it's hard to treat. Why is it so hard to treat, and why is there no cure yet?
So I feel like the number one reason for that is because we don't understand the why for the disease. So we know that it causes neuronal death, the death of neurons. We know that it's because of abnormal polyglutamine repeats, but we don't know what specific interaction causes these abnormal number of repeats. So we know that like there is neuronal death, but what is causing that is our biggest why. And if we can't figure out that why, we can't figure out the treatment on how we want to, you know, cure the disease. So I feel like there's a lot more room to like discuss why this neurodegeneration is happening.
Mhm. What are some early onset symptoms of Huntington's disease?
So that would be ranging from tremors. So you would
Mhm.
start seeing like hand tremors, some abnormal muscular movements. So sometimes you can see patients kind of twitching a lot and kind of losing their balance. That's something I've seen a lot. You also see mood swings and anxiety and depression. That's pretty common with Huntington's disease patients. But I feel like the most obvious symptom is motor related. And people say that Huntington's is the mix of Alzheimer's, MLS, Parkinson's. So it's kind of the intersection of those three diseases, which makes it really, really devastating to watch. But those are some things I've observed. I also work with mice. And unfortunately, I know the use of mice in research is really ethical, but we've gotten the approval for all of that. And when we induce Huntington's disease for the mice by increasing the number of repeats, some of the mice get really, really weak. They fail to walk. So I've seen a bunch of that as well.
Is Huntington's disease present in all species of animals?
Not really. I think just humans, but in order to kind of speed up the research and figure out this big why, we usually use mice for it. I know some labs also use rabbits, but not ours. We use mice. Yeah.
So you talked about the DNMT3A.
Mm-hmm.
How did you figure out that protein and the ELAV1? How did you figure out that they interact with each other?
Great. So basically, we did a RT-PCR, which is a way to amplify segments of DNA to see the quantity of the DNA as well as what are the interactions of it. So we did an assay, kind of an interaction assay for this DNM3TA because that had already been identified by previous researchers that it played a key role in HD. So what we did is through that assay, we figured out a list of more than 15 proteins that interact with this DNM3TA. And with that, they gave me ELAV1. And what makes ELAV1, the protein I work with, special is that it's an RNA binding protein. So a lot of the other proteins are DNA binding. And they wanted to see if maybe the RNA side of things could make a different angle to this big question. So they gave me the RNA binding protein ELAV1.
All right. So you talked about your research, but in your title, the word knockdown is used. Does that mean like a gene is turning off, or is there some other meaning?
Right. So the knockdown is basically turning off the gene. So in order to do this experiment, we needed to knock down ELAV1 because we wanted to see if ELAV1 is knocked down, does the neurodegeneration occur? Basically, just in simple words, so if ELAV1 is gone, we have DNM3TA. But our hypothesis is that DNM3TA and ELAV1 work concurrently to cause this neuron loss. So if we knock it down and there's no neuron loss, that means that ELAV1 plays a role. But obviously it's not cause and effect. There could be so many other factors that don't result in the neuronal loss. But that was our hypothesis, and we needed to do knockdown to ensure that ELAV1 and DNM3TA work concurrently for this neuron loss.
How has your understanding of your field evolved through the process of conducting this research?
I think for me, I'd never done wet lab research before. Actually, I have done a little bit of wet lab research in high school, but that was during COVID and it was kind of hybrid, so I didn't really get that hands-on experience. So I kind of saw molecular biology as something that was linear. So like one cause, one effect, one protein, okay, you knock that down, okay, you know that protein is part of the hypothesis or part of the reaction. But I realized that it's so much more complicated than that. There's so many other factors involved. The central dogma of like transcription, translation, DNA replication, there's so many like tiny players involved that it's not a clear-cut way of doing. Like I thought science was pretty clear-cut, but I just realized it's so much more complex, requires a lot of patience, and requires a lot of critical thinking because once things go wrong all the time, like I've had a lot of experience of, experiments failing and I've had to troubleshoot and kind of ask my mentors. So just learning that or like building that skill of critical thinking and reaching out to your mentors to ask for help, I found that really important for me. Initially, I didn't have that. So growing that over the years kind of helped me grow as a researcher and kind of, feeling like you're a part of the lab. Initially, I was like, I'm just a student, what can I really contribute to this lab? Like, I didn't really have the background, have the knowledge of these PhD researchers. Like, how can I come in and kind of make a difference in this lab? But kind of shifting out of that mindset of, I can grow into like a strong researcher if I really put the skills, the time, the effort into it. So kind of thinking of it that way kind of transformed my view of research, specifically wet lab molecular biology research.
So going on to some of the issues that you faced during your research, what would you say were the hardest problems that you had to overcome?
Sure, that's a great question. I feel like I've had a lot of problems over the years. I actually worked full time at my lab last summer from about June to August, and I feel like that's when I really felt like, okay, research. I'm knee deep into research because usually it's within during the semester, and during the semester I'm balancing like classes, I'm balancing extracurriculars, my social life, my academic life. So I never really felt that I could get continuous training. But last summer was when I had 6 weeks of 9 to 5 research. And that's when I started to realize, wow, like a lot of things can go wrong when you're a researcher without people to like push you back up. So I would say for me, I use this technique called Western blotting. That's kind of to quantify and to see the interaction of proteins. That's what Western blotting is used for. And I initially performed a lot of Western blotting myself. So I got trained my freshman year and kind of that evolved to me performing it independently. And one thing about Western blotting is you need to be really careful. It's a really time consuming process, about 4 hours in total. There's different steps to it. And what would happen is if you're not careful with looking at the time, the band can like go under the gel. So what would happen sometimes is I would just wait 2 seconds longer than I was supposed to wait, and the band would already go past the gel. So the result of that would be a really blurry band for me to kind of interpret. So I feel like that was my biggest struggle at first. It's a mix of like a technical error and a critical thinking error because I could still make sense of it, but I was confused as to what does this really mean. And with that came a lot of self-doubt. I'd be like, wait, am I like really thinking about it? So what really helped me to come out of that mindset or come out of that shell was to book constant meetings with my PI, also known as a principal investigator. Each lab has one. They're the ones who kind of started the lab, they're the one who lead the lab, and they're the one who manages all the grants needed to do the research. So I would book meetings with her and kind of go through my research, my results to see, am I viewing this correctly? Am I making a mistake in how I read the data? So kind of talking things through with her made me understand that, okay, even though like something might have gone wrong in this one Western blot experiment, she can help me walk through it and kind of show me the right way to do things. And I feel like that was a big step for me at first because I kind of felt embarrassed to go to her because I was like, is she going to judge me? Like, is she going to think that I don't know what's going on in the back of my head? We had a lot of other undergrads in my lab, so I was one of four. And they seemed pretty knowledgeable about everything. I think that's because they've had previous research experience. So I'm like, if they know all this, am I expected to know all of this? So it was a lot of imposter syndrome at first. But my main message is like know when to ask for help and definitely ask for help because we don't have the formal training like everyone else, but we do have the interest and the desire to kind of get to that stage. So always be open to ask for help and you like just keep your mentors close.
And if you had like unlimited resources, time, money, equipment, what's one major thing that you would change in your research or like you would add to your research?
Wow, that's a loaded question. So I talked about how gene expression and knockdown is the new thing for HD research. More specifically, epigenetics is the field that's up and coming, which talks about, gene expression, gene knockdown. That's the biggest thing. So honestly, that's the direction I would go in if there was unlimited resources, time, money, because RNA sequencing and RNA knockdown is a very time-intensive process, and there's usually no clear-cut answer. Because if let's say you have four lanes in a gel, two of them seem to be effectively knocked down. but the other two seem like half and half. You don't know if it's knockdown. There's always a 50-50 debate in RNA like knockdown research that you need to use clues and use stuff like between the lines to figure out, okay, do I think it's really knocked down? So there's a lot of questions. So I feel like with the money kind of investing into like some company that focuses primarily on RNA expression, RNA knockdown, RNA sequencing, that's the biggest direction I'd like to go in. And kind of another thing is I'm not the biggest fan of working with animals. So kind of figuring out if we can use AI or computers
Mm-hmm.
to kind of model animals instead of using the poor mice to like do this research.
Yeah.
I understand that it's like there's no other choice we have right now if we want to go somewhere, but like I don't know, now that we have AI, maybe building a model of the brain or building a model of mice and seeing
Like
how those
animatronic mice.
yeah, animatronic mice, something like that to, you know, advance HD research because
Mhm.
we truly need it. I feel like it's been a long time coming and having those advancements can really shape the field. Yeah.
Mhm. So going on to the time management, how did you manage research between classes and extracurriculars and whatnot?
Yeah, that's a great question because at first that was non-existent. Like
Yeah.
I think I overbooked myself my freshman year.
Mhm.
I started second semester, so February of January 2023.
Mhm.
It was exhausting at first because first of all, location-wise, my lab was located in the medical campus, which is about a 10-minute metro ride from my campus, but I used to take the bus at that time, so it was about like a 30-minute commute. So, after I'd get out of class, I would schedule it more in the evenings because most of my classes were late morning to afternoon classes. So I was like, I don't want it to clash with any of that. Another thing to note for context is our school offers research for credit. So I don't get paid for my research. I enroll in this class called Bio 200, which is basically Introduction to Research. So
Mhm.
they've really, WashU really encourages students to get into research. Like we're a big research institution. So let's say I took about 2 hours of credit my first semester, which translates to about 6 to 8 hours of research a week. So with those 6 to 8 hours, I kind of divided it every other day. So Monday, Wednesday, Friday. But the big problem with that is experiments are usually consecutive days. So that was my biggest struggle because I didn't know the world of research. So I scheduled myself every other day. My PI was like, it would be better if you could come every day, maybe your sophomore year, because obviously she understands that I have a schedule this year. So initially, it was really hard for me to come home at a normal time. So I would come home at like 7 p.m. and be like, now I need to start working on my homework or studying for
Mhm.
quizzes and tests for the next day. Or if I'd have a club meeting in the evening, or if my friends would want to go out for dinner. So, yes, it was hard my freshman year, but I took that as kind of like a learning board to revamp my semester sophomore year. So that's when I spaced out my research. I did it back to back. I had more time in my schedule. My schedule was more flexible. So I would go in the evenings of Thursday and then go Friday mornings. So it would be more consecutive and I could independently do a lot of experiments myself. Um, so that really helped my schedule get back into shape. I could dedicate my time evenly, and the credit system really helped me balance my hours. And my lab, I feel like when you're searching for labs, it's really important to find that the lab team kind of fits your personality and your like
Mhm.
way of life or doing college, because my lab was really flexible in the sense that they literally gave me a month off to study for my MCAT, which is a medical school exam.
Mhm.
So kind of finding a lab that aligns with your schedule, I would say, is the biggest bet. Obviously, it's so hard because getting a lab itself is a lot of work, but kind of keep that in mind as you guys are navigating, searching for the right lab for your future research.
Mhm.
Yeah.
Now, being at WashU, which is like such a research-heavy institution, were there any other like peer research projects that really like sparked your interest or that like helped you, focus or motivate you to do your research?
That's a great question. Um, Yeah, surprisingly, I'm like now thinking about it, reflecting on all the classes I've
Mhm.
taken. There's always a research element within every class. But honestly, the most surprising to me is freshman year, I had to take this class called college writing. And the specific field I chose was technology and selfhood. And for that class, our end result was actually a 10-page research paper.
Oh
And I
wow.
was like, English and research papers. I
Mhm.
never saw that coming. So I was like, this is interesting. I'm not the biggest fan of writing. So I was like, okay, I'm going to take this one step at a time and see what's happening. And honestly, it's pretty much the same process of, you finding a thesis, you finding evidence to back up that thesis, you looking at literature, doing a heavy literature search, and kind of piecing that all together to make that final research product. So I feel like they kind of instill that mindset of research early on, because this was before I started my wet lab research at the Yano lab. So it was pretty interesting to see how they bring in research, even when it comes to English and college writing. Yeah. I'm like trying to think about what my paper was about. I think it was about Netflix. I think it was about I think it was about the environment, how binge watching could be bad for the environment. Yeah,
Mm,
that
mm.
was it. So it was something
Interesting.
along the lines of that. But yeah, I think that's the first time I did a formal research project in college.
And for like our audience or someone just starting out in research, what is the single most important piece of advice you would give them?
I would say embrace the uncertainty. Like, it's a really vulnerable thing to be in research because you're never going to get the results you want. Maybe eventually, yes, but when you start getting into it, it's going to be really hard to expect results. Like I said, I thought I started this project 2023. Now it's 2020 and almost the middle of 2025, and there are significant results, but there's no definitive answer as to whether the interaction between DNM3TA and ELAV1 causes the neurodegeneration. So it requires a lot of patience. But I would say don't get discouraged by all these negative results. Sometimes I'm like, wait, is my work meaningless? Like, what am I doing? Just studying one specific protein on one specific disease.
Mhm.
But think of the grand scheme of things, to think about long term. This, even though I'm going to leave the lab in December because I'm graduating and moving on to other institutions, what's keeping me going is this one result, or not even a result, this one set of experiments could inspire the next researcher to build off of it and kind of discover the actual why for HD and what's causing the protein interaction. So think of your project, your personal project given to you, or if you've been helping a PI, because I started off helping my PI, and that moved on to me helping a postdoc, and that moved on to me working independently. Just know that you are not going to get the results you want, and that's okay. Seek your mentors, seek feedback, ask for feedback. That's like the single biggest thing for me that I had to learn, and never give up. That's really corny, honestly. Like
Mhm.
when people say that, I'm like, what? That's hard to follow through. But just know that you're in it from a purpose. The purpose should be from within, because like HD is part of my family, so that really wanted me to have the motivation to find this why. So find your personal why, and you know, just seek advice from mentors.
Mhm.
Yeah.
And you talked about how your advice was like, when you have a setback, don't get discouraged. How do you deal with setbacks?
Yeah, that's a good question. I think I briefly mentioned this before, but at first, when I couldn't really understand my results, like I would do an experiment, because an experiment, you have a protocol, and
Mhm.
following a protocol is like cooking. Like you just follow like the list of instructions for
Mhm.
cooking. So
Right.
that's pretty straightforward. Obviously, I've had to ask questions about where's this located or where's this box located. But that was pretty straightforward. But for me, my biggest setback was not knowing what the results meant. So I would get the results, and initially I would just go straight to my PI, be like, these are the results. But I had like a wake up call a few months into research, and my PI was like, you realize that once you become independent, you can't always come to me to analyze your results, right? So
Mhm.
that was kind of the wake up call. She's like, as a researcher, you need to build critical thinking skills. You need to, look online, look at papers, look at how they analyze results. So that's when I realized, how do I deal with setback? I can't go always running to my PI to answer. I need to take that agency in myself, use my own brain, and kind of figure out what these results really mean. And honestly, I learned it through my PI. It's not like this was a realization that I got myself. So again, my biggest feedback is talk to your mentors. They've been doing this for years. They've had so much experience, and they're always willing to give you advice on how to approach setbacks.
Through your research, were there any misconceptions that you had prior to going into it that you learned about through it?
Yes, I feel like for me, I always knew, maybe not always knew, but later in high school I knew I wanted to go into medicine, not necessarily research. Because I thought research was just, being on your laptop 9 to 5, looking at like n number of research papers
Mhm.
and just analyzing those research papers, finding similarities to that, or just repeating the same experiments over and over again in your bench desk. And like, I don't know, I just had a perception that people would go crazy in this job because you're hyper focusing on one thing and going really deep into one thing. I was like, how is this possible? How do people have the balance between reading literature, doing your experiments, following through with what your lab wants you to do? So that was like my biggest thing coming in first, but that completely changed because I saw that everyone I've talked to in my lab, we're a pretty small team, about 6 of us, 3 senior researchers, 1 postdoc, and a few undergrads with our PI. I learned that collaboration was really important. So I had this image of like a lone person sitting in a bench, just pipetting all day. But it's so much more than that. You consult your colleague for advice on how they approach this experiment. You approach your PI to see if she has any insight about what like solution to use or what reagent to use. So it's a lot of collaboration. It's a 50-50 between laptop work as in literature work versus wet lab work. It's not solely one or the other. It's like a blend of the two. But it's grant writing, that's one thing I never expected. I didn't know that sometimes postdocs write grants themselves, and undergrads too. To apply to my summer research program last year, I had to work on a grant, and I would say that was a pretty challenging thing for me because kind of putting your research in simple terms is the hardest thing to do because you're in your lab, you're always discussing it in terms of, DNA methyltransferase versus ELAV1. So kind of not dumbing it down, but kind of explaining it in simpler terms to the judges or to the people who know biology but don't know my field. I think that was a great learning experience, and that really showed me that researchers should be great teachers because it's impossible to advocate for your own research without explaining it to people who aren't experts in your field. So that was the biggest thing I learned. Yeah.
Yeah. This is a really good talk. Yeah, a really good podcast. Not only for us, probably for our audience too. Thank you so much for joining us.
Yeah.
Yes, thank you.
Yeah. Thanks.